In the first 4 episodes of this series, we've learned about dominant and recessive traits, DNA, DNA sequencing, which revealed the DNA code, deciphering the DNA code, and then editing the DNA code with CRISPR. Now in the big finale, we're going to learn how to use all of that knowledge to cure sickle cell disease in actual human people. Sickle cell disease is when your red blood cells aren't nice little round circles, but long and brittle. Kind of like a sickle. This causes all sorts of problems like anemia and bouts of intense pain. But what is sickle cell disease? Like, what's actually causing it? Well, it turns out sickle cell disease is really interesting in the world of DNA because it's a recessive trait caused by a single point mutation on the HBB gene. To help explain what that means, welcome back my photorealistic drawing of a person. As of right now, they do not have sickle cell disease. But I'm gonna play malevolent God, zoom into their body, and give my photorealistic drawing of a person sickle cell. Here we go! Inside of every photorealistic drawing of a person, you'll find veins. Inside of every vein, you'll find red blood cells. Inside of every red blood cell, you'll find hemoglobin proteins. Now, hemoglobin proteins are the molecules that actually grab onto oxygen, so your red blood cells can efficiently move the oxygen all throughout the body. They're very important proteins. And each hemoglobin protein is actually made up of 4 smaller proteins, 2 alpha globins and 2 beta globins. Your body makes alpha globin with this bit of DNA called the HbA1 gene. And then this bit of DNA called the HBB gene creates beta globin. Now, if I want to give my photorealistic drawing of a person sickle cell disease, all I have to do is change this single A into a T. Out of all 3.3 billion DNA letters in the human genome, sickle cell disease actually is caused by that one single change in the DNA code. And it actually is that one A. This isn't like random code for illustrative purposes. This is the HBB gene. I got it off the NLM website. Now let's zoom back out and see how that one little change cascaded into sickle cell disease throughout the rest of the body. Our mutated HBB gene still creates beta globin, but with one important difference. Now it has a chemical sticky spot. That creates sticky hemoglobin proteins, which stick together and make these long hemoglobin sticks inside of the red blood cells. Those long sticks stretch out the red blood cell into a sickle. The sickled red blood cells cause traffic jams inside of the veins and die much faster than normal red blood cells, causing a lack of oxygen throughout the body and sickle cell disease. All from that one single letter.
Hold on a second. If this disease is really as simple as you claim it is, why haven't scientists CRISPR'd it away? The whole last episode was about the wonders of CRISPR editing. So just change that T back to an A and voila. See, but I know that lots of people still have sickle cell disease. And that doesn't add up. You're clearly not giving us the whole story.
Well, you're right. Sickle cell disease is an excellent candidate for CRISPRing, but the logistics of actually doing it in the real world are really complicated. Let me try to explain as fast as I can. As everyone already knows, every cell in your body has a full copy of your DNA code, but actually, not your red blood cells. They're just bags of hemoglobin. They don't have DNA. But the cells that make your red blood cells do have DNA. Those are called stem cells, specifically hematopoietic stem cells, and they are located in your bone marrow, aka the inside of your bones. The DNA in those cells is the DNA that matters. Like, yes, all the DNA inside of all your cells has that sickle cell mutation, but in those other cells, like your brain cells, they don't make hemoglobin, so it doesn't matter if they have that hemoglobin mutation. Your hematopoietic stem cells are the cells that make hemoglobin, so that's the DNA we need to edit. And like I said before, those cells are located in your bone marrow, which is very inaccessible. Like, remember, with the fish, we had to inject the CRISPR into each individual cell, which we can't do when the cells are inside of your bones. Luckily, those stem cells can actually be removed from your body with a crazy drug that releases them into your bloodstream, so we can take them out and do the edit in the lab like we did with the fish. But There's a big difference. In the fish, we were knocking out a gene to make it stop working. That's actually a lot easier than making a specific edit, and that makes sense. It's easier to mess up the DNA than it is to make a precise change. That's why some scientists are going about this in a different way, by knocking out a different gene that stops a different kind of hemoglobin, fetal hemoglobin, from being produced as an adult. So if we knock out that other piece of DNA, then fetal hemoglobin goes back into production. That's the fetal hemoglobin strategy. But the other strategy of changing the T back to A on the adult hemoglobin gene is also being done, even though the CRISPR process is harder that way. But let's assume that if we've done all of this properly and we have a vial full of successfully edited stem cells with A's instead of T's, now we have to reintroduce those new stem cells back into the body and replace all of the old stem cells. This is called a bone marrow transplant, which has been around for a long time, but in this case, the donor cells are actually your own stem cells that have been edited with CRISPR. Bone marrow transplants are an established way of fixing sickle cell disease, but sometimes it can be hard to find appropriate donors. So in that one specific situation, this whole crazy CRISPR process actually would be a viable option. I also don't want to gloss over the horrific reality of a bone marrow transplant. You're trying to replace all of your hematopoietic stem cells, which means you have to kill all of your old stem cells with chemotherapy drugs, which creates a problem because those cells didn't only make red blood cells, they also made white blood cells, aka your immune system. Once you kill all the old stem cells, it will take roughly 6 months for your new stem cells to repopulate your bone marrow, which means you won't have an immune system for roughly 6 months. If you make it through all that, this whole procedure does seem to work, so hooray, confetti. But be aware you will still be able to pass on sickle cell to your kids, because remember, every cell in your body has a copy of your DNA, and that includes your sperm and egg cells, aka germline cells, which we did not edit. We only edited the hematopoietic stem cells. Scientists don't do germline edits in humans because those edits will be passed down through the generations in perpetuity. We don't know enough about DNA editing to know what the consequences of that might be. Plus, those subsequent generations have not consented to being experimented on, so at least for now, germline edits are a no bad scientist. And that is the current state of affairs for editing human DNA with CRISPR in the real world.
Alright, I must admit, after all the time we've spent together, this whole video series, I feel like I do have a vague but satisfying understanding of modern genetic editing. But that being said, Gattaca was better.